Hi everybody, so let's wrap it up. There's only a few more topics left that I need to cover for the DNA Technologies lecture slot. So we just covered Sanger sequencing, which is a standard sequencing approach for when you're trying to figure out what your particular short DNA fragment contains. This particular video that you guys see is one of the many videos available online about how to design Sanger sequencing primers. You're welcome to watch it if you're interested. I also would like to point out that sequencing design, primer design is critical not just for sequencing primers, but also for just any PCR approach. And again, if you just type in primer design into Google, you will come up with a lot of different tools that you can use. One of the most popular tools that people use a lot is to figure out the melting temperatures of the primers. Now, the standard AP or biology 1010 textbooks come up with this little formula for calculating melting temperatures of primers, except that a lot of research has been done for figuring out the actual melting temperature and the actual annealing temperatures for primers that are a lot more sophisticated than that formula. So I don't recommend using the formula to calculate melting temperatures for the primers, aka the annealing temperature, sorry, not melting, annealing temperature for the primer to optimize your PCR reaction. Instead, I recommend websites like this. So this is New England Biolab's TM calculator. Here you can, if you can choose whatever polymerase you're happening to be using for your PCR reaction. Let's say we're going to use fusion, and you can also select which buffer you're using. For right now, it's not important for me to describe what the difference between these buffers are. When you actually get into doing PCR reaction, whether for work or for labs, you will learn how to do so and which buffers you should choose. And then you can type in your primer sequencing and let's say, and on the side here, I just typed in 11 nucleotide primer. Its GC content was calculated. It's with between 40% and 60%, which is good stuff. And then its approximate melting temperature of only 33 degrees Celsius was calculated. It's way too short. So hopefully this is a helpful sort of little window into tools available for DNA, for PCR, for getting your PCR to work. NMB has a bunch of other tools available as well in their interactive tools function that you can browse through, and some of them we might use or at least explore later in the course when we continue covering other DNA technologies available in molecular biology. All right. So watch this video if you'd like. But now I would like to focus on a different type of sequencing. Sanger sequencing is wonderful for sequencing short DNA fragments. For sequencing very large DNA molecules, it's simply too slow. So the first few genomes, aka the total DNA content of an organism, were sequenced using Sanger sequencing. So the fly genomes, the yeast genomes, etc. But as the Human Genome Project got underway, new technologies were developed in order to tackle the enormity of the human genome. It is 3 billion base pairs. It took about between 10 and 15 years, depending on who counts, to complete this task. And the cost of the sequencing of the human genome using, in some level, Sanger method, and then later on the shotgun sequencing approaches, that were still in their developmental stages. It took about $1 a base, so $3 billion to sequence the human genome. However, the contribution to the field of sequencing of DNA that the Human Genome Project gave was enormous, because some of the funding that went into, some of this $3 billion funding that went into making the sequencing of the human genome possible also was used to fund small fund development of new sequencing technologies. as a result, a lot of new approaches have been developed. And these new approaches, because they're so high throughput and you can sequence so many bases at once, really radically dropped the cost per base of DNA sequencing, now making sequencing of genomes very much possible. In fact, you can see how low the prices have gone right now. It costs currently, if you have access and a permission of a physician, you can sequence the exome of your genome for as little as $1,000. Exome is basically all of the genes that are actually going to become protein, because only a small fraction of our genome is actually translated into protein. So here are some of the novel sequencing techniques that have been developed in the recent past, such as pyrosequencing, which is described in your book, and I'm not going to describe, but that's a really cool technique. Illumina sequencing, which we'll go over right now, as well as things as sequencing by synthesis and many other new techniques that all the big sequencing companies have been continuously coming up with in order to drop the cost of per base sequencing and make genomic medicine, so medicine based on the genomic sequence of that actual patient, actually possible. So let's talk about Illumina sequencing. Actually, let's just pimp a video by TED about which covers how to sequence the human genome. It's a pretty nice video. I recommend watching it. I'm still gonna go over the details, but there's a cute video you can see. So, the Illumina whole genome sequencing, Illumina is a company based in San Diego. I have a few friends working for it. It's a nice company. I recommend it. So let's talk about the general approach. And now, it's colloquially called Illumina sequencing, but Illumina by now has so many different sequencing approaches that when you go to their actual website, you will be inundated with many, many different techniques they have been proposing by now. Because Illumina mostly creates these giant sequencers, and that's what they want to sell, and they want to sell the best one. Okay, backing up. So the first step of Illumina whole genome sequencing is to isolate genomic DNA and then randomly fragment it. That's really important. So they break the DNA into short fragments, about 150 base pairs or so, because that way they can multiplex the sequencing. They don't need to run one reaction per DNA fragment. They can run a bunch of reactions. So because they're running a bunch of reactions at once, millions of 10,000 reactions per slide. I can't remember. They can't do it in a tub or a PCR tube. They have to do it in on a solid surface where they can mark where they can separate each individual reaction and know what each individual dot of a reaction is, so that no cross-reaction occurs. So here we go. This gray stuff is the fractured DNA, the fractured genomic DNA of the genome about to be sequenced. These purple and pink stuff are adapters. So the adapters are ligated to the DNA fragments, and the rest of the technology, both the ligation, the attachment of the DNA strands to the glass plate, and then the amplification of the DNA strands, and then the actual sequencing reaction will be using these adapter primers, which contain a known PCR, a known primer sequence, so you could attach primers to it. So the adapters are really important, also because in the end you will be assembling many short stretches of sequence into one whole sequence. All of these adapters also carry barcodes, allowing the company to tell apart which sequence came from which dot. And by what I mean by barcodes, they have a very particular ACTG sequence built in, which doesn't function as for annealing, but it functions as a stretch of DNA that functions just like the barcode in your grocery store to identify something. Okay, So step 1 was to isolate genomic DNA and break it up. Step 2 was to ligate primers to the DNA. And then step 3 is to use these primers and base pair the primers to a dense lawn of primers that have been glued to a glass cover slip. Then I have skipped a few steps here, but the end result of this preparation is that each individual DNA fragment that have been attached to the glass is then amplified, and all the other identical copies of that same DNA molecule are attached to the are attached around the original DNA molecule, creating a dot or spot, something that you actually can see under a microscope. And then the actual fun begins. So unlike Sanger sequencing, Illumina sequencing also uses terminators, but unlike Sanger sequencing, the Illumina sequencing terminators are reversible. You can attach a terminator nucleotide to the growing molecule of DNA, stop the reaction, record what the outcome of the reaction was, and then first bleach the fluorescent nucleotide, removing the fluorescence from it, and then attach the OH group at the 3' end of the nucleotide, allowing for a new nucleotide to be added to the same strand. So if you guys remember that Sanger sequencing, there was you created a mixture of molecules of a different length. In Illumina sequencing, your molecules are going to be growing, your spots are all going to be growing, and they're all going to be the same length. And you don't have to segregate the Illumina sequencing reaction on a gel, because instead you use a microscope. So just like in Sanger sequencing, you put in fluorescent nucleotides, some go green, purple, blue, or yellow. If the nucleotide has incorporated itself into the DNA, growing DNA, it will be allowed to fluoresce. It will fluoresce purple, and you will be able to see on your glass cover slip that there's a purple fluorescence occurring in this particular location. So a T, in this cycle of the reaction, a T has been incorporated. Next, the fluorescence if the fluorescence of the purple nucleotide is quenched, the H group is converted back into an OH group, allowing for incorporation of a next nucleotide, in this case the blue A. The spot glows blue, that is recorded, the camera will photograph each cycle, and then the process repeats itself. And you can see here that based on what color each individual spot on the glass cover slip glowed, you can tell what nucleotide was being incorporated in that particular spot. So here we have T, then A, then C, then G, then another G, then a T, and so forth. While this spot has C, 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 and then A, G, and say T. So all of this information, the photographs are analyzed by the software of the sequencer and converted from these colorful images, colorful dots, into this actual sequence. And this is about how one of these Illumina glass slides looks like. It's a microfluidic slide, so you can see the teeny tiny lines here because different solutions can be flown over all of these attached DNA sequences, and the solution's easily changed. So even though Illumina whole genome sequencer is not very efficient, you will only get 150 base pairs for each DNA molecule you have incorporated. You can do about 3 billion different strands on a glass cover plate, and they're really small. So the latest achievements of Illumina DNA technology was 1.8 trillion base pairs in 72 hours, which is exceedingly fast. So if you wanted to, you could sequence 600 complete human genomes in 3 days and under $1,000 per genome. However, not everybody has access to these wonderful machines, and you do have to have a $1,000 per genome, and usually, since they can run so many reactions at once, the reactions are pooled and then sorted out later. So about the sorting out, the file that comes out of the machine for each glowing dot is recorded, and the fluorescence is converted back into nucleotide sequence, and then the nucleotide sequence has to be stitched together to assemble the actual genomic sequence. This is done in big computational clusters, and it takes quite a bit of time. So it's actually much faster to sequence than it is to then stitch together the 150 base pair fragment of a typical Illumina reaction. Now, what I wanted to talk about here is that all of the sequencing that has occurred in the last 10 years, the human genome and all other genomes that have been sequenced, have all primarily been funded by public money. So it's your taxpayer money that has paid for all of this. And thus, it's publicly available. So, for example, you can see the entire human genome and you can browse it using the Santa Cruz Human Genome Browser. This is the gateway link that you can see, and this is the approximate output that you get. For example, here I typed in oxytocin, which is a small peptide that we produce that makes us feel attachment and love. So it's a very happy little peptide. And you guys can see that the gene for oxytocin pre-peptide is encoded on chromosome 20 at the very end of the left chromosome arm. This red thing is the centromere. You can use these arrows to move back and forth. You will move this line back and forth on the actual chromosome. And here you guys can see that here's the gene for oxytocin, which you can see there's 3 exons and at least 2 introns, and etc. Now, this is much more genomically involved information, which I would like to cover again when you talk about human genomes. But I wanted to tell you that the tool is available, and if you're curious, if you're naturally curious, there's no barriers for you to come and look up how different genes look like actually on the human chromosomes. The other important tool that I would like to mention as well, both for PCR, for creating PCR primers and other approaches, is the NCBI gene database. Here's the link for the NCBI gene database, and let's go to the actual database. Where is it? There you go. So here I just went here. So because we're interested in genes and not the many other databases that NCBI has, we're going to select gene from this drop-down list. And let's look at a gene of interest. So in your homework, I'm talking about a particular bacterial protein. The reason I chose a bacterial protein is because they don't have introns, so their actual gene sequence is going to be how your mRNA looks like. While hopefully you remember from basic biology that eukaryotic genes are broken apart, and we'll talk about this later in the course as well. Let's come up with, well, sorry, I misspelled. There you go. It's good enough. So you typed in oxytocin, and the NCBI gene database gave you a bunch of possible hits. You're not interested. In this case, I'm not interested in the receptor for oxytocin, but the actual oxytocin peptide. See, it's a propeptide because it's going to be clipped down. But it's a very small gene, so it's going to be easy for you guys to see what I want to show you. So let's click on OXT here. And here we go. We have the actual gene file for this gene. And if I, for example, wanted to know the DNA sequence or the RNA sequence of the gene, I would scroll way down. to this NCBI reference sequence area. And this NM reference sequence is going to contain the mRNA of the oxytocin gene, and the NP sequence will contain the protein. So in this case, I'm interested in mRNA. So here we go. And this is the small file the GenBank contains for the oxytocin. There's first all the people who published on it will be listed. It's a long list. And then you will see the actual gene sequence of the oxytocin gene. So if I wanted to design a primer now against the sequence, I would copy and paste this and then follow the instructions that, for example, for Sanger sequencing, I would select one primer from here, or if I wanted to amplify this by PCR, I would select two primers, one using the sequence from here and then another one using the sequence somewhere from here. I'd like to skip the poly A, not too specific, somewhere probably right there. But that is reverse and complement from what I just highlighted. So hopefully this will help you guys with the homework question that addresses this. I showed you how to find oxytocin. You can find the gene from Caulobacter crescentus. If you have any questions, please email me. Back to work. All right, so one last topic I want to cover is the fact that you can use PCR to produce genes to produce something new. The approach is called fusion PCR. In fact, there's many approaches to do this, but right now we're going to just talk about fusion PCR. So say we wanted to make human cells have green nuclei, green fluorescent nuclei. The gene for green fluorescence is a very famous gene which is ubiquitously used in biology to tag a lot of stuff, and it's the green fluorescent protein. Here you guys can see the actual barrel of the green fluorescent protein. It's basically 12 beta sheets arranged basically in a little tunnel, a barrel, that allows the protein to hold its own fluorophore in a particular location and then glow. It will absorb blue light and then emit as fluorescence green light. So if we wanted to take this GFP protein and attach it to the nuclei of these human cells, say HeLa cells, we would have to take a nuclear membrane protein from the HeLa cells and fuse it to GFP. So it's very similar how the fusion polymerase was made. Remember that earlier today I talked about how the new, really good DNA polymerase for PCR called fusion is actually a fusion of the PFU polymerase from deep vent archaeobacterium, and a DNA binding domain that allows it to bind DNA better. So it's a fusion of two proteins as well. So how can one do this? How can one fuse two DNA sequences? Well, because PCR, because with PCR we have freedom on how we design our primers. We can create primers that not only will bind the sequence of our choice, but also have extra areas that that are not binding the first, that are not binding DNA in the first reaction, but will become useful in the second one. In this case, we designed PCR primers. Here they were just black, but here they were black and say red. And for this PCR reaction, these two PCR reactions were done in separate tubes, one tube and another tube. In this PCR reaction, the reverse primer was just green, but the forward primer contained both the red sequence and some of the green sequence that would allow it to bind the green template and amplify it GFP efficiently. And both of these red stretches are now novel sequence that has been incorporated into the final PCR product of the first round of the PCR reactions. And both of these red sequences are identical to each other and will base pair. Now, in the second PCR reaction, after we have cleaned up and cleaned away the DNA template and the primers and then unused nucleotides using a column, a PCR cleanup column, we can then mix these two products and perform a second reaction. because we were smart and we made sure that these red stretches of DNA are long enough to anneal to each other during the annealing step of the PCR reaction, then during the annealing step, the primers for this second PCR, one of them is green and one of them is black, will bind here in these two locations. But because the melting temperature is also perfect for the red DNA fragments to associate, they will associate as well. As a result, as the PCR, as the Taq polymerases extend these stretches, they will make a fused DNA stretch that is twice the length of the two original templates, and will allow for the will make the reaction, this fusion protein product possible. Now, if we insert this product back into human cells, we will get something beautiful. So you guys can see here that these HeLa cells have now been labeled, the nuclei have been labeled in green, and you can watch this nuclei divide. There's some residual fluorescence in the cytoplasm, but most of the green GFP fluorescence you can see has incorporated itself into the nuclear envelope of the nuclei. There you go. There's just several cell divisions you guys can observe. So being able to create something new using PCR is incredibly powerful technology, and hopefully I will have enough time in subsequent lectures to talk about other applications of PCR, because there are millions of ways you can use PCR in molecular biology. Well, maybe not millions, but at least a good 1,000. So this concludes the DNA Technologies lecture. It's impossible to cover everything for DNA technologies, and I only touched the surface of things that you can do with good molecular biology techniques. If you guys are super curious and you're trying to follow a career in molecular biology, either as a technician, a graduate student, a research scientist in a lab or at a company, I highly recommend learning as much as you possibly can, because there's a lot of new techniques out there, and techniques get refreshed, get optimized and improved every year. But in any case, this is a brief summary of the topics I have covered today, and if you guys have any questions, email me, and otherwise I will be writing the quiz now. Have a good night.